Hello, everyone. I'd like to begin the Kratom presentation. My name is Kelly Peterson, and I am the Vice President of Technical Services for RotaChrom North America. RotaChrom Technologies was founded in 2010 when two scientific inventors started product development that would reform chromatography. They developed the world's first industrial-scale centrifugal partition chromatography platform. Since then, RotaChrom has expanded into a multidisciplinary team of more than 60 people. This includes world-class PhD chemists such as the speaker today, chemical engineers, software control experts, and many other technical and business professionals. Arpad Kansal is the lead scientific officer at RotaChrom. RotaChrom instruments have revolutionized purification processes by providing high purity, high capacity, cost-effective, and environmentally conscious chromatography solutions to researchers and extractors all over the world. RotaChrom has customers performing research and industrial scale purifications in North America, Europe, and China, across a variety of industries, including the pharmaceutical and botanical industries, as well as academia. Year after year, RotaChrom has improved their products, worked on new purification methods, helped our partners in laboratory design, and performed equipment maintenance and service. In the past months, RotaChrom researched, developed, and tested a new purification method for Kratom. This work created a significant amount of data, and today Arpad is going to summarize and explain those results over the next half hour. Attendees can submit questions via the chat form throughout the presentation. These will be addressed after the presentation in the Q&A session. With that, I would like to introduce Arpad Kansal, Lead Scientific Officer at RotaChrom Technologies. Take it away, Arpad.
Thank you, Kelly, for this nice introduction. Ladies and gentlemen, thank you for joining us today in this webinar. The subject of today's talk is Kratom, and I'm going to present you our latest results in the research and purification of this unique and popular medicinal plant. First, I'm going to briefly introduce the plant, the chemistry and biology of its alkaloids, and the recently skyrocketed scientific and social interest that surrounds it. Afterwards, I will analyze the shortcomings and discrepancies in the downstream process of Kratom. Based on these considerations, the liquid-liquid chromatographic capabilities and know-hows that were developed in our research lab will be presented in detail. CPC methodologies of ballast removal, fractionation, isolation, remediation, and also polishing type of purifications will be highlighted. Finally, these identified revenue streams will be evaluated and summarized. So, commonly known as Kratom, Mitragyna speciosa is a tropical evergreen tree, primarily found in Southeast Asia. Both the plant and its products are referred to as Kratom in Thailand or Kratom in Malaysia, and considered as a widely consumed ethnomedicine for its stimulant and opioid-like effects. Its leaves are chewed or boiled to prepare beverages that are consumed to treat diverse maladies, including fever, diarrhea, and cough. Kratom has been used also to enhance productivity, combat anxiety, and manage opioid dependence. So let's jump into the chemistry. The active chemical ingredients, the compounds responsible for the pharmacological effects of the Kratom, belong to alkaloids and possess tetracyclic indole or pentacyclic oxindole skeleton. The major abundant compounds are mitragynine and its structurally closely related isomers, speciogynin, speciociliatin, and pinantine. Here you can see the structures and the corresponding relative proportions in the total alkaloid content. A total of 54 alkaloids have been described in the leaf drug to date, which make up only 1 or 2% of the crude leaf powder. Important to note that Kratom contains the 7-hydroxy derivative of mitragynine, which is extremely potent opioid agonist compound. This is as a safety of concern. As the trend on both of these charts clearly demonstrates, both the scientific and the social interest in Kratom has jumped exponentially over the past 5 years. I would say this is a huge hype. A recent estimate of use in the US showed that around 3.3 million people consume Kratom on a daily basis as a recreational product, often to treat chronic pain, opioid use disorder, and as a remedy for anxiety and depression. These therapeutic effects suggest potential clinical value and are under pre- and clinical investigations. However, I have to mention that no Kratom-derived products have been approved for use by the FDA. Nevertheless, it is fair to conclude that Kratom alkaloids bear a huge therapeutic potential. It should be also added, however, that a lot of research and standardization are still needed to safely exploit this huge potential. I believe that the very first step in this process should be taken in the downstream processing of Kratom. Downstream methods should be significantly developed, rationalized, and controlled. So ultimately, it means that a strict regulation and standardization is needed in this field. Let's just consider the recently revealed fluctuations in the quality and potency, adulterations, and contaminations in Kratom products. Not to mention that only 14 out of the 54 alkaloids are commercially available, which is heavily hampers the medicinal chemistry and the toxicological research. It means that even the chemical characterization of Kratom is incomplete. So in conclusion, we at RotaChrom saw an unmet need in the purification of Kratom alkaloids and initiated therefore an in-house project to develop centrifugal partition chromatographic methodologies which can serve as a robust and efficient platform in future Kratom research and production. But before jumping into the separation applications, let me highlight our product portfolio to you. Starting with the 2-liter rotor volume RCPC, this is the pilot scale machine of RotaChrom with patented and unique cylindrical cell design, to the industrial scale device ICPC with industrial scale purifications, and the integrated and fully automated solvent system handling solution PCPC, we provide large scale, easy scale up, and tailor-made CPC solutions to our customers. For further technical details, please visit our website or contact our sales department. I forgot to mention that all of the following studies were conducted on RCPC at pilot scale. So what can CPC do in Kratom purification? I would say a lot. Thanks to the flexibility of our core technology, different purification scenarios such as ballast removal, isolation, and remediation can be cost efficiently accomplished on the same CPC platform with different solvent systems. Using crude mitragynine extracts, isolates, full or broad spectrum mitragynine concentrates can be produced. And most importantly, I would like to underpin that all of the highlighted separations were validated on RCPC at the multigram levels. In addition, we have also developed the related analytics on LC-MS to be able to resolve the complexity of Kratom extracts and support preparative separations by quantitative data. Here, this analytical HPLC chromatogram obtained at high pH shows a typical LC-MS fingerprint of a crude extract, I would say a typical CPC input material. So major compounds such as mitragynine, speciosiliatin, speciogenin, and pinantane eludes in this region. And more polar minors such as hydroxy derivatives and N-oxides can be found here in the more polar region of the chromatogram. I guess it's obvious to the audience that this capability is a must in any standardization efforts or in quality control. So based on these methods, more than 100 Kratom samples have been successfully analyzed in our lab. Okay, let's move to our first so-called baseline CPC method. In this case, we focused on the selective removal of ballast materials beside the valuable alkaloid content. In other words, in this study, we tried to efficiently enrich the alkaloid content of a crude extract. As you can see on the RCPC chromatogram on the left-hand side, that the resolution of the run is excellent. And I have to tell you that it was achieved by a strict pH control. It's also obvious that a nice throughput can be realized at pilot scale with this method. The change in the physical appearance of the input substance is also spectacular. See the photos in the right-hand side. Important to note that the utilized solvents are class 3, simple and cheap. CPC operated in dual mode. It means that the elution and extrusion periods were applied. And it secures a reliable and continuous production. Moreover, as the mass balance chart of the same run demonstrates here, full alkaloid recovery, see that 10 grams of alkaloids goes into the CPC and 10 grams is recovered from the CPC, can be accomplished with the alkaloid profile preserved. It means that the ratio of the color bars, the composition of the sample remains the very same after removal of the ballast compounds. It means that chlorophyll and waxes are selectively removed during this process. And in numbers, using a 25% mitragynine extract with a 40% total alkaloid content, concentrated, very potent 83% mitragynine alkaloid content full spectrum extract or product was obtained. And such a, it's interestingly, this method is capable to produce also salt-based concentrate and also alternative products such as mixture of these analogous compounds of mitragynine can be simply obtained by simply adjusting the fraction collection times. And the most important achievement in this application, I guess, is that it's fully validated in pilot scale production, and also the cost calculations and rentability calculations are also available for this full spectrum workflow. Our second methodology was intended to fractionate the major isomers of mitragynine, namely pinanthine, speciogenin, and speciosiliatin. As you can see, in the pilot scale RCPC run, we managed to enrich these compounds from a crude, and the RCPC fractions were subjected to flash chromatographic polishing at 100 milligram scale in this case. It must be noted here that the full structure elucidation, I mean the high resolution mass spec and the NMR study confirm the validity of this process. So this process produces high purity isolates at 100 milligram scale. And these isolates can be used as a reference compounds in bioassays or as standards in analytical tests. By the way, these isolates were tested for blood-brain barrier permeability at our partner group at the Semmelweis University. The third method developed at RotaChrom aimed to tackle the safety issue related to the uncontrolled amount of 7-hydroxymitragynine. In this separation scenario, we focused on the selective removal of this highly potent agonist, also called the bad guy in Kratom, from this alkaloid mixture. To put it simple, we succeeded to optimize the solvent system in which the concentration of this compound can be efficiently modulated. In practice, by this CPC method, non-compliant, I mean extracts with 7-hydroxymitragynine content higher than 2% of the total alkaloid content. So such samples, Kratom samples, can be remediated in a simple fashion, resulting in a so-called broad spectrum product. And last but not least, I would like to call your attention to a very challenging separation task that we faced during our Kratom-related research. Since high purity, it means API-grade compounds for clinical studies cannot be obtained by conventional crystallization in Kratom, chromatography is needed as a final processing step to remove some of its critical impurities. So the toughest one is in case of mitragynine is the compound of corinanthidine. Differing only by a methoxy group on the aromatic motif of mitragynine. So it's resulting in typically a high cost, low recovery PrEP HPLC purification. In contrast to this, I'm proud to share that we found a selective solvent. Here you can see on the HPLC chromatograms, just compare the top and the bottom one, a selective solvent. So we found a selective solvent by which the resolution for this couple of compounds, mitragynine and corinanthidine, the selectivity was hugely improved. And I have to mention that the adoption of this selectivity trick, so to say, is still in progress. So coming to the summary, I hope that I managed to demonstrate to you in this webinar that Kratom is an extremely hot topic, and we at RotaChrom gathered significant expertise on the downstream processing of this medicinal plant. I believe that the presented applications and case studies for different revenue streams highlighted the versatility and robustness of our CPC technology. Although I didn't touch in this webinar, the feasibility of these separations was also considered, and the calculations along with the solvent systems and corresponding mass balances are ready to share with inquiring customers. And finally, I would like to emphasize that this work was accomplished in strong collaboration with academic and industrial partners. Last, I would like to mention that our Grow Together program is still open for potential collaborators. The scientific contribution of two Hungarian academic research groups from the Technical University of Budapest and the Semmelweis University is highly appreciated, as well as the Kratom material support from two of our industrial partners is also acknowledged. And personally, I'm really grateful to my colleague Kristóf Gazda, who led this Kratom project in our lab. With this, thank you for your kind attention, and I'm happy to discuss any questions or comments in the Q&A session. Kelly, please.
Wonderful. Now we're going to move on to some questions. First question, what about other separation technologies in Kratom purification? Can you compare or comment on supercritical fluid chromatography or flash LC versus CPC technology? Arpad?
Yes, of course. I would say that a myriad of separation techniques is utilized nowadays in Kratom research and purification in Kratom processing. But I see that the liquid-liquid extraction, for example, coupled with the column chromatography, is a really labor-intensive process, or flash chromatography are also successfully applied in lab-scale isolation, only in lab-scale isolation projects. And I would add that UPLC, GC, and SFC, supercritical fluid chromatography, are typically used for analytical purposes, and prep HPLC, so the highly efficient prep HPLC, is typically described in polishing type of separation scenarios. But nevertheless, as I demonstrated, CPC can cover the whole array of separation tasks in a cost-efficient fashion. And I'm pretty sure that CPC is more feasible than multi-step liquid-liquid extraction to produce highly potent full spectrum extracts. So practically, this is the sweet spot of CPC. I mean, in terms of throughput and operational cost, CPC is really optimal for such purposes, so fractionation of natural products. And I guess even prep HPLC can be challenged by CPC in the case of unique selectivity, in the case of unique selectivity providing solvent systems, and also in CPC is superior to prep HPLC in terms of throughput.
Very nice. Thank you, Arpad. Okay, next question. Is it possible to isolate minor or even subminor alkalides from Kratom by CPC?
Yes, it's a great question. So CPC is what we learned in the last year in this project, that CPC is capable to fractionate and enrich minors in a straightforward way. However, to get really high purity isolates, I mean compounds above 99% purity, a second technique should be included, and it is practically flash chromatography. So in this tandem, so to say, tandem workflow is really widespread in the industry, and we conducted also the same. And yeah, let me just mention that subminor N oxides in Kratom were also successfully isolated with a similar workflow.
Very nice. Thank you, Arpad. We have a question from the audience. Juan is asking, have we mentioned special solvent systems? Can you describe the solvent systems that were used in the separation of Kratom?
Yeah, so the exact composition of the used solvent systems is confidential and can be shared with the potential customers. But typically we use ternary solvent systems with strict pH control in Kratom related methodologies.
Understood. Another question from the audience. They're asking if we've encountered artifacts such as oxindoles during the separation studies of Kratom. And then they're just asking if the instrument is run under inert atmosphere. Arpad?
Yeah, so some degradation we observed during the purification studies, but these degradations or degradation products were really minor ones, so negligible. But the question is really valid, such as N oxides are. I'm afraid that such degradation products cannot be avoided if you don't use inert atmosphere.
Thank you. That provides a good segue into the next question. What about solvent residues? Do CPC solvents remain in the product after the separation?
No. In normal case, no. So with proper drying, solvent residues shouldn't be a problem. And anyway, it should be checked regularly by appropriate GC analysis. And yeah, so our Kratom products were when we use CPC in a production campaign, so our products were fully compliant for solvent residues. We use normal drying ovens, and it was fully compliant. But an interesting observation was that mitragynine isolates tend to form inclusion salts with methanol, so methanol should be avoided in mitragynine polishing.
Very nice. Thank you, Arpad. There's another question that ties into this. The audience is asking if RotaChrom used any additives in the RCPC, or was it solvents only in this separation?
Yeah, as I already mentioned, in the ballast removal application, so in that solvent system, we modulated the pH. So yes, the solvent system was acidified, and the end product was extracted from the product fractions to get removed or to get rid of salt forms of the alkaloids.
Understood. Okay, on to our next question. Does CPC allow users to switch from research on, say, Kratom back to a production mode, for example, work with cannabinoids?
Yes, I would say yes. So it's really easy to switch from research scale to production scale. Basically, it's just the matter of the loading capacity of the system. Yeah, so switching between solvent systems, it's really easy and straightforward.
Understand. So CPC technology allows users to pivot to a different method without a lot of expense or labor. So that's very interesting. All right, the last question that we have is, can you please elaborate, Arpad, on the cost benefits of CPC in Kratom purification?
Yes, but I guess this question is a bit difficult to answer since, as I already mentioned, the composition of the solvent system is a confidential data. But nevertheless, since our methodology is silica-free and utilizes recyclable or at least reusable solvents, I believe that the cost structure of such applications, such purifications, is much more favorable than a conventional prep HPLC methods. And I wanted to add here that supercritical fluid extractions or chromatography could be a choice, but in the case of basic compounds such as Kratom alkaloids, it's not suitable to make it happen. So I would say that the ballpark range for applications such as ballast removal and isolation is approximately a $200 to $400 per kilogram input material. So this is the in numbers.
Okay, very nice, thank you. Another question from the audience. How stable are these alkaloids, either as free bases or salts?
Again, in our work, especially in the production campaign, when we made the toll processing to get this highly potent full spectrum alkaloid mixture, we didn't observe any significant degradation of these alkaloids. But yes, they tend to be unstable in different conditions, but in regular and in typical purification conditions, they should be stable.
Very nice, thank you. Well, really appreciate your research and time today, Arpad, and thank all of you for attending our RotaChrom webinar. We look forward to seeing you again soon at our next scientific presentation. Please visit our website at www.rotachrom.com for more information or to contact us with any questions regarding CPC technology. Thank you all, and with that, we will conclude this presentation.
Thank you, Ken.